My name's John, and this is my research partner, Chang. We're here to talk about an important plant disease called Pierce disease in grapevines. By studying this disease, we hope to develop better mechanisms for understanding and working with these plant diseases. It is caused by a sap-feeding insect that infects the plant. When the plant becomes infected, then tylose form inside the plant, and ultimately results in shriveled berries and vascular occlusions, which, if severe enough, can kill the plant.
And this kind of disease, called vascular disease, is becoming more and more severe recently. It's not only the problem for grapevine, it's also become a very severe problem nowadays in Wisconsin area. Just like oak wilt or Dutch elm, they both vascular disease. So by studying the mechanism, we can get a better understanding to control the disease.
Why don't you show me a little bit about your poster?
And in the figure A, we can see how severe the disease can be. And in the figure B, we can get a closer look at the bacteria in the pores of the water pipes. They're rod-shaped bacteria.
Oh, alright. So to better understand this, we need to first understand exactly what the inside structure of a grapevine looks like. So if you look at figure A, it'd be the same thing as looking straight down a grapevine if you cut your finger and look down it. In figure B, it's the same thing except looking at its side. On the side here, you can see the way they get interconnected through these tiny vessels and different cells. And then, it continues on to how the actual occlusions begin.
When the plants are healthy, the water pipes are open. We can see the pores through. And when the plants are infected, the water pipes are blocked by some material. Most of the time, they're tyloses. Just in the figure C, we can see the longitudinal section of the water pipe. They're blocked by the tyloses. And once they're blocked, the water stops moving, transporting into the plants. Sometimes the vessel, the water pipes will block by the gel or crystal.
And then so that's a general overview of exactly what this disease does to the plants and how it goes. But our research in particular focuses on the super tiny walls that interconnect between these guys. So by looking at these, you actually zoom all the way in to see these down here. And when you see these, at first they're smooth and not roughed up or anything like that, which allows membranes and stuff to be kind of controlled when they pass through. But then eventually, there's later stages of the development.
Firstly, the surface will become rough. You can see here, compared to this one, it becomes much more rough. And then the gel, they will secrete gel-like structures, forming the networks structures. They're connected all together. And sometimes the membrane will bulging from the pores of the water pipes. Once it's forming the porous structures, the tyloses can grow into the water pipes, and so that block the water pipe.
So once they become infected enough and all that porous actually happens, they fully break through the walls and completely occlude the vessels, which is what ultimately causes the death in these grapevines, and which is the reason why we've been studying it for so long and so in-depthly, because with this disease affecting these different plants, all these different varieties of diseases as well, it's very valuable and important for us to go over this, because without this, you're not going to understand it, and without understanding it, you're not going to be able to develop some kind of improvement or way to combat this problem. Good?